Hey everybody. So for this next video, I just want to first start with a very similar beginning I did with the last video, and that is if you click on review here, highlighted, you can watch the crash course video that discussed the resting potentials and action potentials of a cell to help you better understand these next couple of slides. Also, keep in mind that we did actually talk about this in some detail back in chapter 9. So please go back to chapter 9 if you want to see this in a little more detail. So with that said, I'm going to be talking about this kind of like a little summary of what we already know, a couple reminders, and let's start here with the resting membrane potential. So resting potential of a neuron are polarized. So we had a whole video on the polarization of a cell, meaning that at resting state, we're going to see a charge of about negative 70 millivolts. So our cells are going to be a little bit negative. So that said, we have the sodium-potassium pump, which is going to contribute to this resting membrane potential. And this is going to account for about 70% of the energy required for the nervous system. So an electrical charge is called a potential because it has the potential to make charged particles move. So again, if you are polarized, whether it be positive or negative, now you have a charge. That charge is going to attract the opposite. So in other words, we're slightly negative. Those positive ions are going to want to move towards you. So you have the potential to make those charged particles move because you are more negative. And that's really important for an action potential. And then if you look down here with local potentials, so a local potential is a small deviation from the resting membrane potential that is caused by a stimulus, but it may or may not be effective. So with that said, we need to keep in mind one really, really, really important piece of information. And right here, that is that an action potential, can be either excitatory or inhibitory, meaning that if there is an action potential generated, it may want to excite that neuron, or it'll make the neuron less likely to be excited, which means it's being inhibited. So that's really important to keep in mind. So we have two things we can try to do: excite it or inhibit it, make it work or prevent it from doing so. Really important to keep in mind. It'll make more sense in the coming videos. So again, resting membrane, if you look at this graph over here, we begin at about negative 70 millivolts, so we are slightly negative. And that's primarily because of the different proteins that are located within the axon and within the cell. So we remain a little bit negative. If we are looking at the axon, what we're going to see is sodium is going to naturally want to diffuse into the axon because of the lower concentration of sodium on the inside of the axon, and then the potassiums are going to naturally want to diffuse outward. However, with the sodium-potassium pump, we're going to see the opposite. This is going to be an active process. So sodium is going to be removed, and potassium is going to be pulled in. Now, you're probably wondering, they're both positives, right? Sodium and potassium are both positive ions. But if you look here, the trade-off is 3 to 2, meaning we're going to remove 3 positive ions to gain 2 positive ions. So if this were money, for every $3 you spend, you get $2. You do it enough times, you're going to realize you're losing some money. And that's what's happening here. So you're losing 3 and gaining 2. That's a net result of -1. So the inside of this axon will begin to get more and more negative. And it's the same thing we're seeing here. If you have any questions on anything to do with these action potentials or resting potentials, please let me know.